Good morning, everyone. Um, this webinar, it should last 30 to 45 minutes. Okay, so for today, what I'm gonna talk about is the differences between imaging and metrology. So what is the difference between imaging and metrology and comparing different techniques? So I'm gonna compare AFM to SEM and see where maybe SEM is a little bit better or where AFM is actually better than SEM. I'll give you a little background about how an AFM works, and And then I'll talk about some available modes. Now, this is not an exclusive list, but just some common modes that are used. One is called phase contrast, which looks at differences in material stiffness. The other is another one is called KPFM, which you can study oxides or surface potentials. I'll go into some force spectroscopy. This is a way to quantitatively measure nanomechanical properties of materials. And then I'll go into something that's kind of unique to nanosurf AFMs. That's called fluid FM. And this is, it's hard to describe in just a couple words, but it includes nano manipulation, nanolithography, and things like that. And finally, I'll talk about what a good sample prep is. Okay, so what are the differences between surface imaging and metrology? Well, surface imaging is creating a two-dimensional representation of a three-dimensional surface. So the X and Y are encoded with dimensional information, but the Z is qualitative. So an optical image in an optical microscope is a good example of this. The Z in an optical microscope is just isn't represented by anything, really. Surface metrology is creating a three-dimensional representation of a three-dimensional surface, and the X, Y, and Z are all encoded with quantitative dimensional data. So when you're talking about an SEM, comparing an SEM and an AFM, an SEM is obviously surface imaging, so is an AFM, but if you're talking about surface metrology, an SEM doesn't have that quantitative Z dimensional data, whereas an AFM does. So what is it? I mean, I think everyone's familiar with electron microscopy. An SEM is qualitative. For example, these four samples here all have different topographies, but we can't quantify the two differences in the roughness because electron microscopy doesn't allow for true Z measurements. So image contrast in an SEM, for example, depends on electron surface absorption or scattering. It doesn't depend on anything else. So if you had materials that absorbed electrons, more electrons than not, it might look darker, right? And you can't say that that's lower in height. Also, SEM doesn't work well with insulating samples. And the lateral resolution, this is true for also optical microscopy, is coherence limited, meaning it's limited to the wavelength of your SEM. So in KV or whatever. So atomic force microscopy, this is a near field technique. So we use a sharp probe to scan the surface. So with this, what that means is the resolution is just limited by the tip radius. So if you have a 2 nanometer tip, your XY resolution is 2 nanometers. It also we can measure the Z axis, so we can create a 3D rendering of the surface. It's suitable to use with hard or soft samples, insulating or conductive, air or liquid. So you have a lot more range of type of samples that you can image. And this is an image here on the bottom left is an image of a Flex-FPM. Here on the right are two example images. One is a double-stranded DNA image. This is a 200 by 200 nanometer image. You can just make out the double-stranded, it's a double-stranded DNA, but you can just make out the double helix in each of these little ridges here is the double helix of the DNA. So we can see a very high resolution. On the bottom is a 7 by 7 nanometer image of mica, and you can actually make out atomic lattice of the mica. So comparing the two techniques between SEM and AFM, in SEM you're measuring in a vacuum, AFM in ambient. For controlling temperature, I mean, there are stages or cryo or heating stages that can go down from -100 to about 1,000 degrees C in an SEM. In AFM, you're kind of limited to a smaller range, but between -35 and 250 C. This is a little bit about the way it works, maybe in ambient conditions versus anything else. Sample prep, you need a conductive coating for SEM, and for AFM, there's very minimal sample prep required. You don't have to modify the sample at all, really. And I can get into that at the very end. Liquid imaging, yeah, it's possible, but not really. Not really many people do it. You can't. Some people are, but with a lot of pain. AFM, there's no issue at all. Just put the sample under liquid, start imaging. In a controlled environment, or there are environmental SEMs or variable pressure SEMs, we can also, for an AFM, we can use sealed liquid or gas cells, or even operate the AFM in a glove box, so with a controlled low oxygen environment. Now here's something that maybe an SEM may excel at. They have much larger samples, 300 by 300 by 300 millimeters, 12-inch kind of wafers or larger. With AFM, if you have a tip scanning AFM, which is what the Flex-FPM is, then you are really unlimited by the sample size. You can place the AFM directly on top of a sample and image that directly. With other designs, other manufacturers, it's kind of system dependent and sample size dependent. So key advantages. SEM gives really nice images, so best at imaging. Pretty flexible with sample size and shape. They're easy to use. And there's a lot of other detectors that you can add. So you can get chemical info, crystallography info, structural info, etc. AFM, well, it has the highest resolution for X, Y, and Z. You have additional modes that are just not only topography, but you can get mechanical, electrical, magnetic modes out of an AFM image. And you can also operate the system in ambient or liquid conditions. Well, disadvantages, well, you have to operate an SEM in a vacuum. If you have a sample that charges, that absorbs electrons, you can get just a little bit of struggle with the image quality in that. It is destructive to sample, so you can damage actually the sample itself. Not only that, but if you need to coat the sample with something conductive, like a layer of gold, etc., obviously that's destructive to the sample. And you don't get the true surface metrology, meaning you don't measure the actual Z height of a sample. Some disadvantages, well, you have a limited scan volume of the AFM. So our scan area is limited to 100 by 100 by 10, or it's actually probably 12 microns for the Flex, and then you have to move the sample around. I mean, obviously with SEM, you can get a much bigger field of view with the scan. It's considerably slower. It probably takes, for a good AFM image, 3 to 4 minutes, maybe longer for a single frame. Or obviously in SEM, you can get a frame in a couple seconds. And the last is the potential for scan artifacts. So if you pick up something with the tip, because we're scanning with a tip, with a sharp tip, you might pick something up, or the tip might get blunted, and then the shape of the feature you're trying to image will look different. So where does AFM excel in the imaging and metrology space? So I've added optical here. So this gives you basically the range of horizontal features, the size. So with some optical techniques, you can get down to below 100 nanometers. If you look at super resolution techniques, or if you're looking at sort of like white light interferometry, things like that. And vertical dimensions, they start at about, is that right? Yeah, 1 nanometer for maybe interferometry techniques. SEM has a little bit higher resolution in X and in Z, and obviously AFM gets the highest resolution in X. So we saw that atomic resolution, the lattice resolution, and sub-angstrom Z resolution in Z. So here, for example, to give you that sub-angstrom resolution, we have piece of HOPG, so this is graphite. It has, when you pull off a layer of it, it can stack into multi-layers. And so here's 4 different layers of it, and between A, B, the lower half and the top part, there's about 2 nanometers, 2.5 nanometers of spacing there. But now we can actually see the layers between A and B. There's a 330 p, so 3 angstrom. One atom. We're measuring the height here between A and B and C and D. That gives you a indication of the resolution of AFM and how low it can go. So how does an AFM work? The simplest case, the cantilever has constant deflection. So if we're away from the surface, so here's an image of a cantilever and tip. The laser bounces off the end of the cantilever and hits a detector. And we try to align the detector so the laser hits right in the middle of the detector. And this was what we call no deflection in the cantilever itself. Below the cantilever, we have a surface. As we push the cantilever against the surface, the cantilever will bend. That will cause the laser to deflect to a set point. And this is the set point we set in the controller, in the software. Then all we need to do is scan the surface, so drag this cantilever across the surface and maintain this constant set point. In order to maintain that constant set point, we raise the entire cantilever up and down to maintain constant force. So that's the simplest case of an AFM. The first AFMs were built this way. There's another mode which is actually more popular. It's less destructive, called dynamic mode or tapping mode or non-contact mode, or whatever you want to call it. And that also leads to something called phase contrast mode, which is the first extra mode I'll talk to you about today. So in dynamic mode, instead of excuse me, the cantilever being not moving at all, we take a small piezo at the base of the cantilever, a shaker piezo, and we send an AC voltage to it, which is called an excitation voltage. And this gets the entire shaker, the entire cantilever to start shaking. And the cantilever is like a diving board. So the end of the cantilever will start to oscillate. And now, so that laser that's bouncing off the end of the cantilever, instead of hitting a single spot on the detector, is also moving up and down on the detector. And we form an AC kind of signal from that. So this is what's called the free vibration amplitude. So when we're far away from the surface, we might have a certain amplitude of in nanometers, maybe 100 nanometers or so at the end of the cantilever. As the cantilever approaches the surface, what happens is that it starts to feel the surface itself, and so that amplitude gets dampened. So it reduces in amplitude. And so what we may have started at 100 nanometers of amplitude, we might now be at a 50 nanometers in amplitude. And this is what we set the controller at the set point. And we tell the controller, I want to scan the surface at this 50% of the free vibration amplitude set point. And again, it raises the cantilever up and down to track the topography of the surface by maintaining this amplitude. The benefits of this is that it's intermittent contact, so you're not always in contact with the surface. You can preserve the tip, you can preserve the sample a little bit more because you're not physically probing the sample at all times. And it leads to extra modes that you can get. And one of the modes you can get out of that is called phase contrast mode. So we have the same kind of setup here, but this signal right here that I get out of the detector, photodetector, has a frequency component, and it has a phase component associated with it. And the phase is just the difference between this excitation voltage and the signal voltage. So the difference between the phase difference between the two. So we use the amplitude of the signal as the feedback, just in tapping mode. That's what we would use. And now the phase signal here, so the difference between these two, excitation and the signal, changes in relationship to the force that the surface or that the tip feels on the surface. So forces can be anything. It can be mechanical forces. electrical forces, magnetic forces, any sort of force, the cantilever will react to that force, and the phase will change according to that. So in phase contrast imaging, first the amplitude is used for feedback. So this is to get the topography. And then we get a second channel, which gives us material stiffness or any sort of force measurement. So we get kind of two measurements at once. The metrology, the Z, the topography, and we also get how different materials on the surface, how stiff they are compared to one another. Now this is it's a qualitative technique. So we'll be able to see some materials are stiffer and softer, but we won't really be able to tell what exactly the stiffness of the material is. So here, for example, is a phase contrast image of let's see, an SBS-PS polystyrene blend. And on the left is a topography image. So this tells you the height differences in this polymer blend. And in the middle is the phase contrast image. So this tells you basically that the lighter areas are different from the darker areas. And we can see a little bit in here, the topography in the middle section here, which kind of is I mean, there are some islands in there, but we can actually see in the phase contrast that those islands are actually of a different are a different material, right? So they have a different stiffness than the area behind. And then along this edge here, we see these dark spots inside of that. So these little islands, dark islands, tell us that that material is different than the surrounding matrix around it. So this is a great way to see how polymers mix. We can heat up the sample and see how if these islands join together and then cool it back down. The image on the far right is a topography image where we've rendered it in 3D and overlaid the phase contrast image on top of the topography image. So we get a good indication of both channels at one point. So other modes that we can do, well, MFM is magnetic force microscopy. We can, remember phase contrast, we can image any sort of force, so magnetic forces. EFM measures electrostatic forces. There's another mode, which I'll go into a little bit later, called KPFM, which allows you to identify the work function domains, so areas of different charge, surface charge. You can do something called lateral force microscopy, which allows you to identify differences in friction, so areas that have less friction or more friction. Force modulation is kind of similar to phase contrast, but it's another way to look at mechanical stiffness, contrast. There are more advanced modes called scanning microwave microscopy, which allows you to measure capacitance, and this is a kind of imaging doping profile of semiconductors. That's one of the main uses. And force spectroscopy, and I'll get into force spectroscopy actually at the tail end of this talk. And that's a way to quantify the elasticity and adhesion of a sample. So first example I'll give of an advanced mode is KPFM. So this is Kelvin probe force microscopy. This is on a sample of graphene oxide. So this is graphene on top of a copper substrate. Then the graphene is oxidized, and so on the left is the oxidized part, and on the right is the actual unoxidized graphene. Yeah. So we get a topography image, so we can see differences between the two areas of the sample, but really we are unsure about what the to get a more contrast, let's say, a bigger difference, what we can use is KPFM signal. So, and what that allows us to see is actually there's some areas of the sample which have either breaks. So we can see that here in this in the blue area here, which show the difference between the oxidized part or the copper oxide and the graphene part of it. There are some differences in the KPFM signal that you can see here, and also here that don't show up in the topography. This gives you an idea of the KPFM has a little bit higher sensitivity to look at electrical differences in electrical mode. So the area here is crystal defects, and then we have differences here in the substrate coupling. Oh yeah, and here in this corner here. So that is information you get, extra information you get that's not available in the just topography image. So it gives you more information about your sample. So we can also measure the contact potential across the copper oxide and the graphene, and just a line cut right across, and we can see actually quantitatively what that value is, the difference in contact potential. So KPFM can be extended to, there's other kind of flavors of KPFM. There's things called, what I was just describing is called amplitude modulated KPFM, but there are things called sideband FM, so frequency modulated KPFM, heterodyne FM KPFM, which is kind of like the thing everyone's talking about now, and some things called pump probe KPFM, where you can actually look how, or basically freeze things in time and look at how the KPFM signal works in time channel. And all these are actually, except for amplitude modulated KPFM, we can do them with some add-ons. So we could either use some lock-ins from Zurich Instruments to look at some of these other types of KPFM signals, if that's something that you would be interested in. So force spectroscopy. So in force spectroscopy, it's going back to this this image where we are in contact mode. If we're just making contact with the surface, so the microsecond before you touch the surface, there's no deflection. If you push into the surface will compress, right? The cantilever will bend, and we'll get a deflection, which represents the force. So deflection is proportional to force, and then we can use the force, the amount of force that we've applied, and the vertical displacement, so how far we've pushed into the sample, to calculate the mechanical properties. So in a typical force distance curve, so we have on the x-axis the tip sample separation, on the y-axis is the deflection, so the amount of force. So as we approach the surface, so we're going toward the surface here, we make contact with the surface, and then there's a increase in the force as we push into the surface itself. We have the baseline deflection, 0 force, but we also have the max force, so amount of force we applied. We also know how much we've indented into the surface, and we also have an indication of where the actual surface position is by the point at which the force starts to increase. So the slope here, well, it's a little more complicated than just a straight slope, but the slope here is related to the elastic modulus of the samples, the Young's modulus of the sample. When you retract, so when you pull the cantilever off the surface, you get a similar curve, but you get maybe some adhesion between the tip and the surface. So we can use this to, for example, map chemical domains on the surface if we had a functionalized probe. So once you do a bunch of force curves and you understand the slope, there's a variety of contact mechanics models. The most common is the Hertz. And this is used with a spherical tip with large radius, and when you don't expect any adhesion, and it has this an equation, etc. I'm not going to go through any of these too much, but there are 20, 30 different contact mechanic models. These are the most common ones here. DMT, JKR. This is used a lot when you have adhesion in your sample, etc. So let me show you some examples of force spectroscopy on some sort of samples, and this is one on rubber. So here's a 4-component rubber sample. The sample was microtomed to get a flat surface, and then we took force curves. So in a grid pattern, at each pixel, we took a single force curve, and then analyzed that using the DMT model. And if you take a look at the actual modulus map, what we get is an image of the actual modulus of this rubber sample, of this 4-component sample. We can see in the histogram of the elastic modulus that it looks like there are 4 domains, and that aligns with what this actual sample is. And so we can actually see the actual 4 domains and the stiffness of this 4 domains. The resolution here, we can see that some of the smaller domains have about a 350 nanometer in size, the XY lateral size. So force spectroscopy is a great sample to use machine learning on. And so for this sample, what I did is a simple k-means clustering, probably the easiest unsupervised machine learning technique. Taking the raw data, this allows us to, if we took a lot of data, allows us to actually analyze it a little bit faster, and allows us to look at things that you might not come up just from looking at just the raw data. So in the sample, which we know we have 4 domains, we can see that using k-means clustering, we have the prediction here, and it gives us kind of the average value for those 4 domains, starting with about 150 megapascals up to 1.3 gigapascals for these stiffer kind of inclusions, circular inclusions here. But what you can see is around the kind of the smaller ones, there is some mixing there. So it thinks it's part of this kind of orangey, salmon colored domain around the edges of it. So we can see here that actually around the edges, it is some mixing. There's some small area of mixing, but with this k-means clustering, we can also get an idea of what the actual percentage of each of the 4 components are, from almost a third of it with this larger salmon colored one to the stiffest one taking up about 17% of the total area of the sample. You can also measure nanomechanical properties of cells, of live cells. So here's a example of a HeLa cell. It was grown on a petri dish in cell culture media, and this was done at room temperatures. But you can also try to keep the cells, happy at a little bit higher. And these are 3 examples of 3 different force curves. So this is an actual height map taken from the force spectroscopy data. But if you just took 4 positions here and then graphed them, what the force curves look like, you can see that there's differences. Clearly, there's differences between the 3 force curves, representing a force curve on the petri dish, which you expect to be very stiff. So we see that there's a almost vertical line there. You have a second force curve that's along the edge of the cell, so it's a little bit softer. So there is a slope here, and then the third force curve that's on the cell body, and you can see that the slope is the least, or is the shallowest, I guess. So this represents the softest of the 3. So we can do the same thing. We do a map. This was a 64 by 64 pixel force map. So I can't do the math in my head, but a lot of force curves which you analyze, and we can get a number of channels, like topography, which is the contact position. Using a Hertzian model or something like that, we can get actually the stiffness, and we can see very stiff where the petri dish is, which is black regions here, but then we can see the stiffness across the cell. We can also get max adhesion force, which doesn't look like there's much difference in the adhesion across the entire sample. Between even the petri dish and the cell itself, there's no difference there. And the other is pull-off distance, which is the distance at which we detach from the surface. So again, we can do some histograms there. So maybe the more interesting one is the stiffness, where we obviously have 2 domains, one representative of the cell and one of the petri dish, which are much stiffer on the far right, the histogram there. And we can also do a 3D representation because we know what the height is, and we can show you how the stiffness of it relates to the height of the sample. So the last topic I want to get into is something called Fluid-FM, which is kind of a unique add-on to the Flex system. The way it works is, so Fluid-FM stands for microfluidic force microscopy. The way it works is that you have a kind of special kind of cantilever, which is hollow. So, I mean, it's completely hollow, and it's attached to kind of a microfluidic system which has a reservoir where you can put any sort of media in the cantilever, and it's attached to a microfluidic pressure controller, which allows you to apply pressures, positive or negative pressures, to the fluid that's inside the cantilever itself. So this attaches to the scan head itself. You can put this on an inverted microscope if you wanted to, or you can do a standalone with just a top view. The fluid FM comes in a number of different probes. There's a kind of a tipless style probe with a large aperture up to 8 microns in diameter. There's more standard kind of AFM cantilever style probes which have a sharp tip, but at the end of the tip there's a small aperture up to 600 nanometers across. And then they can also manufacture probes which have a sharp tip but have the aperture in the side here. And these are all done by standard FIB processing. And actually you can even purchase probes that don't have any hole and then use your own FIB to make a custom size design. So the FPM, or so to Flex-FPM, fluid probe microscopy, has a lot of different modes. And I mean, I can talk a whole hour about all the different things you can do with it. Some of the most common are single cell adhesion. So you can pick up cells with it. You grab a large cell, apply negative pressure, you can attach it to the cantilever and then pull it off a substrate. And that lets you measure the cell surface adhesion force, or you can do cell-cell adhesion. You can do the same with bacteria. You can pick up beads and use the bead as kind of the colloidal in colloidal spectroscopy. So this is force spectroscopy with a large bead. You can do cell sorting. You can, with the cantilever I showed you with the aperture on the side, you can inject material into cells. You can extract materials from cells. You can do spotting, so you can do nanolithography or printing. You can deposit material on a substrate, and you can do this in air or in liquid because the cantilevers are completely enclosed. Nanolithography allows you to draw patterns with the cantilever itself. So yeah, so what I wanted to talk to you about, maybe go in a little more detail, is the single cell injection. This is what the cantilever looks like. At the end of the cantilever, there's this small aperture. So when you have a force curve, because it's a force sensitive instrument, you can actually figure out where you are in the cell. So this looks like a force curve of an injection, or actually this is beyond injection. But what happens is when you push against a cell, you have a point given by Roman numeral 1, where you actually touch the cell. At position 2, you can see where the cantilever perforates the upper cytoplasmic membrane. At position 3, it perforates the upper nuclear membrane, etc., all the way through until you reach the substrate at position 6. And at each of these, so you can push against a cell, and just using the force feedback that you get, determine where you want to inject into the material. So if you want to inject into the nucleus, obviously you need to be inside the nuclear membrane, so you need to be between position 3 and 4 in the force. So the volumes that you're able to control are in the picoliter range. So you can see the amount that you can inject into a cell, and the smaller the volume that you inject into the cell, the more chance that the cell will survive too. So there's good yeah. So here's an example of transfection to direct transfection to plasmid into a nucleus. So here's a cells cultured on a petri dish. The cells were injected with a GFP encoding plasmid, and you can see that with over 2 days now, the cells start to transfect the GFP and start to glow green. So the fact that they're transfecting means that they're alive. And they found that they can get about 40% transfection efficiency and inject up to 30 cells with a single cantilever. So cell extraction works just in the opposite direction. So you can push positive pressure, push material into a cell. What you can also do is extract material for the cell. So this is if you're trying to do proteomics, if you're trying to extract any sort of cytoplasmic or nuclear material from the cell. And it's such small volumes that you can do this while keeping the cell alive. So the idea is push into a cell, but apply negative pressure, lift off, and then dispenses onto something that you can analyze, either an SEM, TEM, PCR, anything you want after that. So the last bit of this talk for the last 5 minutes or so, or even less, I wanted to talk to you about some sample prep and guidelines for sample prep. So as I've been getting through, the AFM is a surface measuring technique. It can only measure features that are on the surface or outside of a sample, on the top layer of a sample. So in order to do that, the sample must be securely attached to a suitable substrate. So I mean, if you're working a lot with SEM or TEM, you probably already know this. It has to be attached to a stub or something like that. So you can't image powders, basically, or colloids unless they're attached to a substrate or glued down or something like that, or put into an epoxy and then microtome the epoxy. For biological materials, we can measure DNA, cells, or extracellular vesicles or things like that, but they can't be in media. They have to be attached to something. So what makes suitable substrates? So for high resolution imaging, so if you have something very small, DNA, nanoparticles, things like that, the roughness of the substrate must be much less. I shouldn't say less, much less than the height of the feature. So a 5 nanometer nanoparticle or DNA, which is 3 nanometers in height, you can't have a substrate that has a 1 to 2 nanometer roughness. You have to go with something much flatter. So what are typically used in AFM? It's well, the most common is using mica sheets, and these are small sheets that you can reuse. You can peel off the top layer, reuse them over and over and over again. They're atomically flat, and these are a lot of times these are used for nanoparticles or even biomolecules. You can also go out and purchase, or maybe you have them, or a silicon wafers, crystal wafers. A lot of times these are used for 2D materials work. You can make these conductive, so you can do KPFM on them. A lot of this materials is available from Ted Pella, which I'm sure you're familiar with. For Petri dishes, now Petri cells are obviously much bigger than DNA, so the roughness of the cell of the Petri dish doesn't actually make a big difference. But Petri dishes you can use for live cell imaging or force spectroscopy. So you can culture the cells in the Petri dish, make them happy, put them under the AFM and image. You don't need any extra sample prep for that. You don't need to fix them or anything like that. They can be alive. But we need to use low profile Petri dishes only. So these are Petri dishes with a sidewall less than 10 millimeters. That typically they're 12 or higher. So there's a special kind of Petri dish with low sidewalls. You'll also need to make sure that the cells are confluent or spread out along the surface. If they're floating around in the media, it doesn't help. We won't be able to do anything with it. So in that case, if you just have a typical glass or plastic Petri dish, some cells don't like that as much as if you had it maybe coated with collagen or PLL or something like that. So you can coat them with whatever you want to just help the cells stick. Another thing is, common thing is glass cover slips. So again, cells need to be confluent, spread out, and on the surface. And again, use collagen, whatever you need to make sure the cells stick to those cover slips. And it doesn't matter, we can image it in a little drop of liquid right on top of this cover slip or in a Petri dish. It'll be the same. So with that, that concludes my short presentation, just about in time. Everyone have a great day, and I hope we'll be in touch soon.